Yes, thanks to those who woke up. It's true that it is challenging to keep the attention for so long. So yesterday I presented some theoretical work that was vaguely inspired by problems in game theory. Today I will try to tell you something about how we could make sense of the payoffs that we introduced in the models, and in particular how we do so in a system where we can do some experiments. And even though we are not as quantitative as what Wenying showed yesterday, we can get some kind of insight of what is the influence of interactions between cells on their reproductive success. So before I start, I would like to acknowledge two great scientists who have been really influential in my way of thinking about these problems. The first one, you have heard him cited several times already last week because he was also Christian's advisor. And so Carl Sigmund at the University of Vienna, he was the person who introduced me to evolutionary game theory. And the second person is a local, Vidya Nanjundia. I hope I have pronounced the name correctly. He is a professor at IISc, sorry, here in Bangalore. So if you get the occasion to talk to any of them, just grab it because they not only have this beautiful mustache in common, but they also are really exquisite persons. They are excellent scientists, but also very humble, always very open to discuss new ideas. They don't judge people who know nothing about their field, as was my case when I started interacting with them, and they are really ready for sharing. So it's really nice to have this kind of human beings in the academic system, which is sometimes a bit hard to get on with. So, Vidya, sorry, I forgot to say, he's really a world specialist in cellular slime modes. So cellular slime modes are one form of multicellular organisms. Multicellularity has, in fact, we usually think most of multicellular organisms that are unicellular organisms because these are those we see with our eyes, but it comes in very different flavors, and not all multicellular organisms are like us. There are some who have very, very different life cycles. What is found when people try to trace back whether multicellularity has one or multiple common origins in the history of life is that it seems that it has originated more than 25 times and also in different forms. So some forms are colonial, some others have a clonal kind of development. So for this reason, among all the evolutionary transitions that I showed you yesterday, this has been named like a minor major transition in the sense that it doesn't seem to be all that difficult to happen. Contrary to, I don't know, endosymbiosis, it has seemingly happened just once or twice. I mean, depending on if you consider mitochondria and chloroplasts. But anyway, it's not as widespread all over the tree of life. So you can find multicellular forms in prokaryota, so bacteria. You can find multicellular forms in eukarya, in fungi, and so on and so forth. So for this, based on this observation, people started thinking there must be some kind of general principle that favors multicellular over unicellular life that has been played over and again. So the kind of organisms, multicellular organisms that we are used to think of are animals and plants that have a very convenient feature. They all originate from a fertilized zygote, and then the body is predominantly clonal. So all cells in our body carry more or less the same genome. Thanks to this feature, a life cycle where you have clonal growth is very efficient in purging cheaters, if you want to put it in these terms. So let's say you have a lineage that starts growing in a way that is completely unchecked, and then clonal growth that is here illustrated by snowflake yeast. I'll tell you briefly what it is. So if you have clonal growth starting from a single cell, then you can sort of break off the part that is sick and then keep the part that is functional. So this form of yeast was obtained experimentally in the group of Will Ratcliffe, starting from usual unicellular yeast that is maintained in cultures as a unicellular organism. So cells are just do not necessarily stick with one another. They tend to be independent. And now he decided to have a protocol of artificial selection where he was collecting what cells would come to the bottom of the vial after, say, I don't know, 2 hours. And then he was taking only those cells and start the cycle again. So this is a very powerful way to play evolution and selection in the lab. And by doing so, what he obtained is a form of yeast that actually forms what he calls snowflakes. So basically, cells do not necessarily separate at cell division. So he claims that this is a sort of a very primitive way of evolving clonal multicellularity. And it is something that can be easily done. It has been done now in also other organisms. So this goes again in the direction of saying, okay, it doesn't take all that much to go from unicellular to multicellular. And now he has many more results. So these things, he's keeping them in the lab, keep selecting them with different ways. They are becoming, you know, more and more complex and structured. So I suggest you to look up at his work if this is interesting for you. Another form of multicellularity that can be seen easily in this kind of system is aggregative multicellularity. So here you have that cells are maintained in culture. So this also exists in yeast. There is a strain, Flow minus, who is actually producing, or Flow plus, I forgot. Anyway, who is producing an extracellular polysaccharide, basically a glue, and then cells stick together, but they also stick to cells who do not produce this glue. And the result is that in a culture, you may see these clumps, and then you will have that clumps at a certain point break down into parts. And as you can see represented here schematically, this is a much less efficient way to purge for a mutant that would be disadvantageous for the group. So this is not just like a lab curiosity. There are many cases of multicellular or pluricellular pluricellular aggregates in nature where the composing parts are different. So symbiosis, for instance, microbiomes. So there is a variety of bacteria, for instance, who live in our gut, about in the order of 100 different types. And they manage somehow to stay together and to be transmitted together to the next generation. In a more broad sense, even communities could share this property. So you have several different species who live together, do not exclude one another, and to a certain extent, which I cannot go into the details here, but we will see tomorrow, they can also be considered as coherent units. And this is an example from a paper by Will, where you have a picture of flocking and non-flocking yeast that do these aggregates together. So this heterogeneous cellular aggregates are typically attributed some kind of function. So people don't think they just happen by mistake, but that there is some advantage for cells to stick together even though they are heterogeneous. And the first simple function is just size. Let's say you have a number of cells, and if they are alone, they can be predated upon more easily than if they are in a group. And if you want to wait until the group grows by clonal division, it may take a long time. If cells can just stick together, then they will do it, and they will avoid being eaten. Other properties that have been studied in yeast, for instance, in flocking yeast, is stress resistance. So when yeast flocks, it is less susceptible to stress because cells outside are protecting somehow the cells inside. And there are several examples of division of labor in this kind of systems. In the case of communities, so resilience, productivity, there are several features, collective features of communities that depend on their being composed of a diversity of species. And in the case of the microbiome, it is very likely that homeostasis within a host is associated to diversity. So when the microbiome is reduced down to a small number of species, it is usually associated with some kind of sickness, pathogeny. Finally, it is possible, and I guess Wenying will speak more about this, that the coexistence of different species allows to exploit or to produce certain compounds that one species alone will not be able to do. So this heterogeneity for all this function is actually quite important. It's not that important for size. Size, it doesn't matter. You can have a big size of the same type. But then, of course, if you are in an environment where you have cells of different types, the most efficient way to gain a big size is just to put everybody together. And so that could be a bit of an accident that you accept, let's say, heterogeneous aggregates. So as I was mentioning yesterday, this heterogeneity can lead to conflicts, and these conflicts can be resolved, at least theoretically, by providing some assortment between cells that depend on interactions. And this assortment can be active, like I am recognizing the partner I want to play with. This is typical in humans, but for microbes, this is not always that obvious. And especially think about the origin of aggregative multicellularity. So if cells need to recognize one another because they help one another to get some collective function, then this signaling should sort of pre-exist the existence of aggregates. But it doesn't make a lot of sense that a signal that is useful for a collective purpose has evolved before collectives were there. So you get into this chicken and egg kind of argument where you say, okay, you need recognition, I don't know, for instance, kin recognition in order to get collective functionality, but then you need collective functionality in order to evolve kin recognition. So this is something that was bothering me a lot. So in microbes, as I was mentioning, you can have different kind of ways to achieve this assortment. So this is again a yeast, multicellular yeast in Will's lab. This is a bigger one, so after many generations of selection. So cells just stick together, and this we know. There are several species, for instance, of the Volvocalis family, where the fact of sticking together has led to more and more complex multicellular structure. There are also other ways by which cells just happen to be close to one another. This is a very, very nice setup that was developed by Alma Dalcò, who unfortunately was a very young scientist who passed away 3 years ago. And she developed this microfluidic chamber where she had two kinds of bacteria that were auxotrophs, like the experiment Wenning spoke about yesterday. So the green cells need the red cells to grow, and vice versa. And then it turns out that the structure that you get in this colony, that is a growing colony, depends on the parameters of diffusion and of production and absorption of the amino acids that they produce in excess or they don't produce. Okay, this has also very nice theoretical work associated to this. I will not talk about this because, well, this is somehow repeating what Wenning was saying yesterday. So just by this passive special structuring, you can get that when you start with very few cells and lineages get segregated, then you can evolve, for instance, cooperating strains. So this has been done in several systems. What I'd be mostly interested in today is what happens when cells are moving. So cell motion has the advantage that cells can go around and look for their food, but with respect to assortment, it's a bit of a bummer because cells can just mix up. And as we have seen, mixing up is not good for sustaining the evolution of cooperation or of collective properties. So this is a very nice example of Proteus mirabilis from the lab of Karin Gibb. Actually, she's no longer in Harvard. She's now in Berkeley, I think, or somewhere on the West Coast. So she showed that these two different strains are actually able to recognize one another and they sort out specially. This is much less clear in the case of Dictyostelium discoideum, who will be the subject of my talk today, where actually cells that belong to very different strains, or even sometimes even different species, they aggregate together. Here is not very clear, but you have that a certain proportion of cells are fluorescent and some others are not fluorescent. And these two kinds of cells come together in the same aggregate. So this feature of having, let's say, multicellular aggregates that originate from initially sparse cells is not only a feature of this one species. There is actually a number of different species, both prokaryotes and eukaryotes, who have life cycles of this type. And I will detail more how the life cycle looks like for Dictyostelium. So this so-called aggregative multicellularity has evolved at least 6 times independently. And yeah, it has many different forms. So sometimes there is division of labor, sometimes there is not. But in general, all these microbes, when they are starved, they tend to aggregate and to form some form of a multicellular aggregate. This multicellular aggregate, in many cases, undergo a development and results eventually in a diversified fruiting body. That's the kind of question we would like to know. We don't know because nobody has seen this transition, you know, from unicellular to multicellular. So this is where we try to use some models to try and see whether we can understand what are the minimal conditions that are needed to go from unicellular to multicellular. Knowing that in these multicellular aggregates, They are all chimeras. I mean, it's very easy to obtain chimeras. So these are some nice chimeras that we saw last weekend in Hampi, in various temples. You can see this is a lion with a frog's body, and this is a mixture of an elephant, a lion, and I forgot what else. Anyway, this is just to mean that they are heterogeneous sort of by construction. And this is very nice because we can do experiments on them, and it's not as difficult as, you know, if you want to make a chimeric human body. I mean, there are many human bodies that are naturally chimeric, but it's not something that is really easy to obtain. So here is the star of today's talk. Dictyostelium discoideum is one cellular slime mold. There are also acellular slime molds. I don't know, Physarum, perhaps you have heard about this. This amoeba that solves mazes. There were a couple of Ig Nobel Prizes given to people who studied it. So this is, well, here is a paper by Vidya where you can be introduced to every detail of this. But this is briefly the multicellular part of its life cycle. So it's not the only part of its life cycle. So cells, they can just divide in isolation. When there is food, they will just keep dividing vegetatively. They do not aggregate. Each one goes on its own. They can also have a sexual part of the life cycle that is much less studied, and I will not tell you about. But let's say this is what we can reproduce in the lab. So we start with a culture of cells, and normally they eat bacteria, but in our case, we have axenic strains that just eat a medium, culture medium. And at a certain point, we starve them. We take away the food, and what happens is that these cells, when they are put on agar or on a surface, they will start signaling to one another and aggregating. This aggregate has several intermediate steps, but at a certain point, it will turn into a slug. And within this slug, cells are not completely mixed. There will be certain cells that start to be in the head, some others that start to be in the tail. And their position here will determine the eventual development of the fruiting body. So cells that are in the front will end up making a stalk, and cells that are in the back will predominantly make spores. These spores, the spore head gets lifted above the ground. So I forgot to tell you, this is a ground-dwelling amoeba that you find almost everywhere. So this slug migrates to the surface of the ground and then creates this little fruiting body. It's like 1 or 2 millimeters high. So that spores that are resistant to stress, they can get dispersed, for instance, by insects passing. These spores, once they find again an environment that is favorable, they can start their vegetative growth. Okay, I should have put some bacteria here as well. So they need food to grow. Yeah.
Is the fate of each amoeba dependent on which position it occupies in the slug?
Yes,
like
exactly.
So
it is correlated. So there is quite a lot of dynamics within the slug. Cells sort of flow from the anterior part to the posterior part. But in general, cells that are in the front, they will end up being in the stalk. Those that are in the back will end up being in the spores. And there are more details to this. Actually, there are not just spores and stalk that are formed. There are more other cell types. But let's say the most important are these two cell types. So given this life cycle, people have considered for a long time this as a great example of altruistic behavior or altruistic division of labor. And this is because there are certain cells here that are sacrificing themselves. I forgot to say, so the stalk cells are dying. So these cells are dying in order to allow these cells to reproduce. So in some cells, people thought, okay, this is somehow analogous to germ soma difference in animals. So we, most of our cells die, but if we have kids, we pass on our germinal line. So there is a sort of division of labor between germ line and the somatic line. So this looks somehow like this. And just to give you a more, so I'm not exactly sure how to start this, to give you an idea of how aggregation happens. So this is a movie that was taken in my lab. You can see that each little dot is a cell, and cells come together to make these aggregates that are called the mounds. And after a while, you don't see them very well because they go out of the focal plane of the microscope. But then after a while, these mounds transform into slugs. These slugs are very highly phototactic, chemotactic. So there is really a function to this multicellular stage. And in the end, what they will do is that they make this fruiting body that was not visible there again because it goes in the vertical axis. As you see here, if you have some differences in the cell that come together, they can get differently distributed within the fruiting body. Yeah.
Could you explain why the stalk has to die before the bulb starts spreading the spores? Or is it correlated or it's a causation?
So not all species have a stalk that dies. Some species do. I mean, why? I guess that is the question. Why would some cells give up their reproductive success for others? If it was clonal, then that would be like germ soma differentiation. You are favoring your kins. So one explanation is that actually these fruiting bodies are mostly clonal. And therefore, kin selection, you are actually favoring your kins. And there is some evidence of the existence of kin recognition mechanisms, but in my opinion, and I think in Vidya's opinion as well, they are not super convincing. So there are some genes that are called tiger genes that are hypervariable in the wild. And they are attachment proteins. So the idea is that there is like a lock key mechanism that allows cells to sort, to create some assortment in the direction of meeting mostly cells of the same type. But in reality, when Vidya has several papers on this, if you go out in the wild, you take a fruiting body and you look at how many different genotypes you have inside, you will find several ones. So it's not very efficient as a mechanism of sorting. So if the mechanism is there, then one may wonder why didn't they evolve, perfect sorting? Just, they only aggregate with their kins, but they don't. And many of these measures are also a bit indirect. So yeah, this I think this is the question. Why did they evolve this life cycle where some cells die to allow the reproduction of others? So this brings me to one typical interpretation of, or a framing, if you want, a theoretical framing of this problem. So imagine you are mixing two different strains, and one strain you say is a cooperator and the other one is a cheater. So how do you decide who is a cheater and who is a cooperator? Actually, you let them aggregate. you let them form these chimeric slugs, and then you just, so since all these cells will die, you just collect the spores, and then you look at what is the proportion of cells of a given type in the spores. For instance, here, these red cells, they are overrepresented in the spores, and therefore they will be more represented in the next generation of free amoebae. And the idea is that if you keep cycling like this, you should select for strains that produce more spores. And there is some evidence that this indeed happens, at least at the beginning. Strains that produce more spores, if you keep doing this in a lab, in very controlled conditions, you will have that these red cells spread for a while. But nobody went all the way to say that this actually replaced the blue cells. And the existence of absolute cheaters that would be expected here is somehow inferred through some other means. That is basically finding some strains that are really sick, and so they are very efficient cheaters, but when they are alone, they can't even form a fruiting body. Sometimes they can't even aggregate. So these cells are indeed, sort of winning this competition. They're found in the wild, but they have not been evolved gradually in the lab. They can be obtained by some knockout or, some genetic screening. So let's do this, let's make this a bit more quantitative. So in the lab, you can decide what is the proportion of cells of one type that goes into the initial mixture. So let's call this X0. And then we can collect the spores, and now we will have the proportion of the same type in the spores, XT. So this T is usually, for Dictyostelium, like 24 hours. This is the time to complete this cycle. And now if one measures the difference between the proportion at the end and the initial proportion, that is the expected proportion of spores that you would have if there was no assortment or nothing was happening in the interaction between these two strains, then you will get what is called the spore bias, in this case of the red type, when it is mixed with the cyan type. Okay, this is relatively easy to do. And then you can say, okay, when this number is positive, then I will call the red strain a cheater, because it will have the advantage over one generation. And then you say, okay, if it has the advantage over one generation, then I infer that it will always have an advantage. And this is quite critical. So typically, this kind of experiments are done by mixing 50% of one type and 50% of the other type. And the other inference that people usually do is that, okay, if 50% and 50% decide that red is a cheater, then it will be a cheater for every frequency of mixing. You sort of need this in order to use like a game theoretical approach on this. So you want to associate the social behavior, so being cheater or cooperator, to the genotype. Okay? So now let's consider that we have here the proportion of red cells in the population, so with the red genotype. And let's say that this is the spore bias that I have measured. I start from this composition with very few red cells. Red cells have an advantage in the spores. You can report this on the x-axis because this is like the additional bias that you get because of this fitness advantage. And then next generation, you will have more red cells. And if you keep going, you will end up necessarily, according to this framework, into a population that is only made up by cheaters. So it's again the tragedy of the commons, as we saw yesterday. So this profile would be associated to a cheater. What would be the profile associated to a cooperator? Yeah, I saw somebody, right? Can you draw it? Yes. Yeah, somebody else? Draw it, just with your hands. So what is a cooperator? A cooperator is a strain that has always a negative spore bias with respect to a cheater. So it will be something like this. Okay? So this is actually it's not exactly the specular, it's, since I chose them symmetric, you don't see it, actually it's the symmetric with respect to 0.5. And now you can have two other situations that you have already seen theoretically in Chaitanya's talks. What are the two other possibilities? Pardon? Coexistence, and then how would that look like, the profile for coexistence? Okay, the suggestion is should look like, what did you say? You said like this, okay? That's not right. Yeah, exactly. So it needs to be this way, because you need to have a positive spore bias when you are rare, and then a negative spore bias when you are common, and then this positive spore bias is bringing you in this direction, the negative one brings you in the other direction. So, and in this case, you have bistability. So the other case, this negative spore bias is telling you that if you start from here, you will end up in this solution. If you start here, you will end up in this solution. So it turns out that actually, when you measure mixtures of dictyostelium, you most often end up with some kind of frequency dependence. So also this one is frequency dependent, but in the end, classification of the behavior is the same. And these cases are a bit more interesting. Okay, so this is something we can measure. But now the problem is how are these spore bias profiles determined? So if we want to understand how we got there, we sort of need to know something about the mechanism that is shaping these profiles. And remember that these profiles are observed at the end of a series of events. So first, cell aggregation, and then development of the multicellular organism. So it's, there are many things that happen within this time span. So there have been a bit of development in the way people looked at this system over the years. At the very beginning, people thought, okay, everything is determined by the genes. So I have a genotype. This genotype is determining my social behavior. And then I will have a sort of hierarchy between the most cooperative and the least cooperative strains if I take different wild strains. So people took different wild strains, they mixed them pairwise, and they said, okay, there is a hierarchy actually. There are the good strains and the bad strains. Actually, I forgot in which order they are. And this is genetically determined, is not transitive. So there is this hierarchy. So you get a prediction about what strain should win. But then it turns out that people started realizing, actually, if you change a bit the conditions in which you are doing this experiment, it turns out that the hierarchies change a bit. So they are not completely overturned. There are always hierarchies, but it seems that it's not all that clear who are the good and who are the bad. And then, not long ago, so the group of Thompson, so colleagues of Wenning, started to look at the full profile of the spore bias. Okay, this is stock investment is another similar measure. And they realized that actually there is this frequency dependence, and this frequency dependence, from their point of view, can be explained by the existence of a strategic game. So now they don't think anymore to the game as something, a game that is played between strains, as I did before, but they are thinking about a game that is played among cells. So a cell will decide whether they want to become a spore or a stalk, depending on the social environment that they experience. So when they co-aggregate, there will be a point in time where they are in this mound that is not yet very specially organized. And within this mound, there are signals that are exchanged. And therefore, every single cell is able to evaluate more or less what other cells are there, what they will be doing. So they have a game theoretical model that proposes the fact that you get this kind of non-linearity in the payoffs due to this strategic investment. And here, again, the behavior is genetically encoded, but it is genetically encoded somehow like a mixed strategy. So you have some probability of becoming a spore, some other probability of becoming a stalk. Another colleague of Wenning has, so his group has, however, shown that there seems to be some dependence of the probability of becoming a stalk or becoming a spore from the phase of the life cycle, the cell cycle cells are into. So cells in a given phase of their cycle, they tend to be more often in the spores or in the stalk. There's also many studies about this, Matsuda in Japan. So this kind of, if this is the reason why a cell decides to become a spore, then this is not something that can be, highly controllable genetically, because every spore, every cell has to divide sooner or later. So this will be more like a lottery mechanism. The fact that you become a spore or that you become a stalk is actually just randomness. When does aggregation start? And then what is the relation of my phase of the cell cycle with respect to the time of aggregation? So these three different kind of frameworks correspond to a very different predictive power of spore bias. Because as I showed you, so the picture I showed you before, highly simplified, basically corresponds to the case where the spore bias is fixed once and for all by the genetic background of the strains that you are mixing. But if you are here, actually being one strain or the other doesn't really matter. It only matters when you are dividing. Of course, if you have two strains that have completely different life cycles, perhaps this will anyway end up with a bias. But let's say you cannot use the same kind of tools to predict what will be the long-term evolution, and therefore calling cheater someone that after one aggregation ends up being overrepresented in the spores, it's a bit of a terminology problem, but somehow when you say cheater, you associate to that the possibility of invading a population. If it is a cheater who is not able to invade, then perhaps one should choose another name. So we would like to understand where along this axis we are actually with Dictyostelium. To what extent reproductive success is determined by genes or by something else. And here comes the work of a former PhD student, Mathieu Forget, who came up with a way of controlling phenotypic bias in chimeras so that one could sort of weight the genetic and non-genetic part of it. And then we try to understand, to come up with hypotheses about the cell mechanical basis of biases. And to do so, we used some models for group formation. So, and I will end up telling you that since I got some new results last week, I'm no longer sure that all this is completely relevant. So there are some very recent developments. So what Mathieu did is that, so we started from the hypothesis that a way to change phenotype without changing genotype is to take a same culture and harvesting cells at different times in their growth phase. So this is just the growth of a population, you know, from a small number of cells up to saturation, as Wenning told yesterday. So if you remain in the exponential phase, in general, things tend to be pretty reproducible and stable. In the moment in which you start approaching a stationary phase, then it is, yeah, there are many things that change in the physiology of the cells. So if no, it's super difficult to synchronize these cells. We have tried a lot. There are some groups that manage. We tried out all the methods that are published, and we weren't able to do this. But we also use a strain that is not necessarily the same of others. So there's plenty of things that are sort of strain dependent. Yeah. So, yeah. No, I know there are groups who manage to do this. These are not synchronized. However, we sort of expect that there are some phenotypic differences between these different times of harvesting. And so what Mathieu did was that he took two populations that were harvested at different times. So since we had fluorescently labeled cells, we could control the amount of cells of each type that were put into a mixture. So he mixed them. He measured this proportion through flow cytometry, then plated them, collected the spores at the end of aggregation and development, and then again measured by flow cytometry what was the proportion of the spores of the two types. Okay, we call this chronochimeras because they are sort of chimeras, but they are chimeras between cells that are isogenic. So all these cells are genetically identical. They have some phenotypic diversity due to the time of harvesting. So what he found is that by doing these mixtures, the result was changing depending on what was the time of harvesting. So here in both font, you have the focal species, so the species for which we are considering spore bias. And the species against which it is evaluated is the early exponential, sorry, not species, the culture, the early exponential culture. So it seems that depending on when you time the aggregation, you would end up saying, okay, one of the two cultures is a cooperator or is a cheater. And this changes with time. And this is something you would conclude also for cells that are genetically identical. So this is a bit disturbing, but it goes in the same direction as this other papers that are showing that context dependent is actually important. And there is it's a bit problematic to classify a strain as a cheater or as a cooperator just based on the genes it carries. Yes, you don't see. Yeah, yeah. Actually, all this is, so it's normalized with respect to what we mix this, because there is a slight difference in the fluorescence of the two cells, so it is not perfectly a straight line. We wash the plate and then collect with buffer, and then we collect the buffer and count through a flow cytometer. So this is a classic way of doing. So Vidya has done a lot of this. It's done in many labs. So now, perhaps this is just a small variation that means nothing with respect to genetic variation. So this could be phenotypic plasticity, but then if different strains are really different in the way they behave socially, then this might be negligible. So what we try to do is to take different strains. So in this case, we took 2 lab strains that are very divergent. So one of them has a whole genome duplication. So they are, in general, considered to be, not as different as 2 different species, but anyway, they are genomically very different. And it turns out that when they are mixed in the same environment, one of them in general gains over the other. And now what we did is that we either took both of them at the same time, and in that case you can see that there is probably some kind of frequency dependence, not very clear. But then we took one of them in early exponential phase and the other one in late exponential phase, or we swapped, we did the opposite. So depending on what is the time of harvesting, you would classify one strain as a cooperator or as a defector. So this means that this kind of sort of tiny differences, because we are talking about a few hours difference in the growth of the culture, seem to be to give effects, phenotypic effects, that are important also when there are genetic differences. So this is not just tiny variations on top of genetic diversity. So this, however, doesn't tell us anything about why this happens. And there are a few observations that we can make, though. So the spore biases, they reflect some initial differences among cells. So this means that there must be some way information transits from the beginning of aggregation up to spore formation. And as I was saying before, there are many things that happen in between. And we know that, for instance, within the aggregates, when cells start expressing differentially genes, there is a lot of change in gene expression. So how exactly is this information maintained through all these changes is unclear. So an hypothesis that we thought of is that perhaps biases are established very early in the cycle, so at the moment of aggregation. And in that case, that would probably be irreversible for later stages, even though you can have compensation and other biases that occur at the multicellular stage. So in order to explain this, I need to introduce what are loners in dictee, so what people call loners. So these loners are just cells that remain outside the aggregates. There's always about between 10 and 20% of cells that do not enter the aggregate. And this has been treated in the past. So both these papers have some experiments and also some modeling. And in the model, these loners were considered like a strategy in itself. Like, you could have a certain percentage of remaining outside. In this other work that tried to go deeper in that, they realized that actually there is a frequency dependence that is not really compatible with the fact of having a fixed probability of aggregating. And therefore, they came up with another mechanism, another model where they say, okay, cells have an intrinsic diversity in a threshold of sensitivity to a signal. And then when they aggregate, if the signal is like a quorum sensing signal, is above a certain threshold, then they aggregate. If not, they remain outside. So we thought, okay, let's go and look at loners in our experiment. As you see, okay, here all the groups are, of course, they say nothing because we wanted to be able to spot single cells. We can count this. And it turns out that actually the loner's bias is correlated to the spore bias. So conditions where there is a larger number, so the culture has a larger number of loners, corresponds to the case where this same culture has a lower number of spores. Somehow, if you put your cells in the loners, they will not form a spore. Sounds rather logical. But how does this happen? So why should certain conditions be associated to a different number of loners? So Mathieu looked at the motility of individual cells, and he found out that there seemed to be two classes of differently motile cells. Some cells that were hardly moving, and some cells that were moving a lot. And the proportion of these two different types depended on the moment of harvesting. So you had so a higher fraction of slow cells in exponential phase, and then this fraction was decreasing over time as they entered the stationary phase. So cells become more and more motile as you get towards stationary phase. That is where normally you have aggregation. So it sort of makes sense. And this is something that was observed already. This bimodality of motility was observed by Vidya long ago, but they only had a very small number of cells. So they sort of inferred this from the distribution of calcium concentration within cells. Okay, so we were quite happy of this result. We thought, okay, things sort of make sense. Let's say cultures with a higher fraction of non-migrating cells, so those that are in exponential phase, we produce more loners, and therefore we'll have a negative spore bias. So that is what we observe. And another thing that was sort of compatible with what people already know is that non-migrating cells might be cells that are dividing. So when cells divide, apparently they detach from the substratum, so they can't crawl anymore, they stop. And then this is compatible also with another mechanism that was shown in dictyostelium in different conditions, that is go or grow. So you can either decide that you divide your cell, but then you will not aggregate, or you can go for aggregation, but then you cannot divide. So all this sort of made a lot of sense. And the additional observation is that cells in exponential phase divide more often than in stationary phase, and therefore, if cells who are dividing are those who are not migrating, then this would explain the bias. Okay. So now we wanted to use some models. I think I'll skip this because I don't have much time. I just wanted to stress that there's not only motility that is an important physical parameter in this system, but also adhesion. So Sandrine made an evolutionary experiment where she was selecting for adhesion. And it turns out that if you select for adhesion of single cells, so not in their multicellular state, you will end up influencing the multicellular state. You will influence aggregation, and there are some interesting features that have been also observed by this Japanese group of Savai in terms of the spatial patterning during aggregation. If you're interested, we can talk about this later. So I wanted to get some time to talk about models. So we try to understand what would happen if we have a model like the one I showed yesterday, where you have self-propelled particles. And this time, instead of having differences in adhesion, you have differences in speed of motion. Okay, you have again cells that are represented by particles that are endowed with an endogenous speed. So they do not align this speed like in the Vichek model, but when they are close enough to interact, then they will update their speed depending on the position of the distance from the other particle. So we get some kind of grouping, and we can start by saying, okay, what happens in this model? That was, by the way, is basically an extension to two types of particle to the model that was proposed by Szabo long ago to describe the motility of other eukaryotic cells. Pardon? I'm sorry.
The force vector F, what is that?
So this is like the adhesion, and this is the strength of the adhesion force. So by simplicity, we say, okay, there is an equilibrium radius, an equilibrium distance, and if you are within R0 and 0, then the two cells are interacting, and they will be attracted. So the force will be positive, but there will be also repulsion, so they can't overlap completely. So if you choose just one cell type, so the difference with respect to the studies that have been done by physicists of active matter is that now we want to have just a finite time for aggregation, because this life cycle has a certain duration, and that is not extendable as much as one wants. So what happens is that you get different phases. So you have some phases where cells are alone, and they can be alone for two reasons. Either they are very fast, and then you have evaporation, or they are very slow, and within this time frame, they didn't make it to aggregate. And then you have several phases of aggregation that correspond more or less of what our colleagues have observed under different experimental conditions, much more controlled. So you get, in some cases, you get formation of groups. In some other cases, you get these labyrinthine patterns. So now what happens if we mix two different kinds of cells, one that is low and one that is fast? And then so we let them aggregate, and then we take out all the groups, and we look at what is the distribution of, what is the proportion of slow and fast cells in the group. So this is exactly like what I did yesterday. Based on this proportion, we attribute a certain, we know what is the bias, what is the average bias. We average over all the groups. And what we find is that this bias is, in general, is frequency dependent, but when the time that is left for aggregation is sufficiently small, then you will have something of this form. So you will have that slow cells have an advantage when they are rare, and fast cells have an advantage when they are rare. So therefore, you would predict that if you repeat this cycle over and again, exactly as I said before, you would converge to the coexistence of a population of slow and fast cells. Okay, so we were quite happy. We thought, okay, all this is sort of consistent. And then last week, I get this new data from Alexandre Souchard, who has been doing new experiments. So he started everything from scratch. He ordered the same, in principle, the same strain from a library. He did his measures with higher spatial-temporal resolution, developed an automatic tracking method. So here you see the tracks of, I think, more than 7,000 different cells. You can see that, that there is no like systematic bias in the direction of movement, so this seems to be quite isotropic. And now that you have all these trajectories, you can start, asking, are these like bimodally distributed when they are bimodally distributed? And so the first surprise was that under these conditions, which are slightly different from the ones that were used by Mathieu, actually we still see a bimodality of motility based on the mean square distance. So there's a fraction of cells that are super diffusive and a fraction of cells that are subdiffusive. But actually the proportion seems to be a bit swapped with respect to what Mathieu has seen. So he sees a majority of cells that are subdiffusive. So that are, if you want, slow. I mean, the point is that we realize that actually this concept of slowness contains two parts. One is like the linear velocity, and the other one is the persistence of motion. And so far, we had just changed the linear velocity and say, okay, they are slow and fast. But it turns out that actually these two populations have some difference in their average instantaneous speed, but mostly they are very different in their turning angle. So the super diffusive population is actually not turning, it's just going in one direction. So we still have to integrate this into models because it's not at all clear that what the results we obtain by changing linear speed are the same as if we change this time scale of persistence. And it's something we are doing now. Yeah, just the last thing I wanted to say about Alexandre is that he, in parallel to what he was doing in the lab, he developed a very nice calculator for the carbon impact of an experiment. So this is something that is concerning us more and more because we are using a lot of plastic, a lot of material to do this kind of experiments. And I personally, I sometimes wonder, is this worth it? Just we wanted to know, to quantify this. And luckily in France, there is a group of researchers that have started already more than 10 years ago to put together some ways of estimating what is the carbon imprint of some, for instance, consumables of the lab. So what Alexandre has done is that he developed this sort of a calculator where you put inside what you're using in the lab and other information, like, for instance, he lives in Poitiers, that is quite far from Paris. So his carbon budget from transport is quite high. He has to take the train to come to the lab every day. And you can put this kind of information. You can put information about, outside outsourcing and so on and so forth, and of computing power. And in the end, you end up having a certain evaluation of how much does a set of experiments cost. So if any of you does experiments, I really encourage you to get in contact with him and, Not sure, but he's developing this, so he will certainly get there, yes. Because I think this is very useful. So, I mean, if you're writing a grant and you want to have an idea of how much carbon you are allocating to a given scientific question. All right. So I only have a quarter of an hour, so I guess I'll go a bit more quickly here. So what I showed so far are measures that are realized in a system where there is already a life cycle that is very convenient because you start with cells that are scattered. I forgot to say they divide just once after they start aggregation. So you don't have like competition going on during the aggregating, the multicellular phase. Then they interact, you just get the spores at the end. This is great because you have a sort of already naturally an intrinsic time scale at which you are observing the system. However, it is not obvious where this time scale comes from if you think about organisms that were originally unicellular. So you have a unicellular organism, how does its life cycle come about? And there are several people who have started asking about this. This is important because if you want to compute, so this is the conditions proposed by Lewontin for evolution by natural selection that Chaitanya already talked about. So you need to have entities that vary with respect to one another. This variation must be inherited, and there must be some fitness difference that is associated to these differences. Okay, if you want to have the fitness differences, you need to have a parent and an offspring. You need to be able to distinguish parents and offsprings. So if you think about microbial associations, this is not necessarily always true. So with Paul Rainey, we proposed a more general way of looking at things where what is important is that you have a way to recognize aggregates over and again. And then once you're able to recognize them, you can also try to establish some kind of genealogy. I'll go back to this this afternoon. So evolution by natural selection can occur more generally than this in these conditions when it occurs in population of repeatedly identifiable units. But how does this repeatedly come from? So, yes.
You are replacing the above triad by the triad below, sort of for the definition?
So, if you want to evaluate inheritance, you first need to have a genealogy. You need to know what is the parent and what is the offspring. But if you don't have a well-established parent-offspring relationship, you can still have correlations between, you know, a group at time t and a group at time t prime. But then you need sort of to know what are these times, t and t prime. So what is, how do they recur? There are many different ways groups can turn into groups at a later time. So this is a bit of a conceptual, you know, paper. It's
where I'm just a little bit of confused. Is it like the variation will go to identification, inheritance?
No. No, but I mean, let's say that this tree satisfy this, so it's a subset. Okay, so a very nice idea that came from Benker and Paul Rainey. So Benker was a former colleague of Wenning, is that actually you might have that ecology is providing the fuel to generate life cycles. For instance, let's imagine that you have a cheat in a social context, and this cheater is actually the only one that is able to survive to a later time and is able to turn again into a cooperator. So if this is true, and this can happen by genetic or non-genetic mechanisms, then you can have, in principle, the emergence of a life cycle where you alternate cheaters and cooperators. And this was then turned into an experiment in the lab of Paul, But this experiment was, so it didn't go all the way as this model, because somehow the timing of the cycle was imposed externally by the experimenter, just to make this more amenable to experimenting. So somehow the time scale of this life cycle was imposed, and this idea that you can have an external time scale that sets the pace for this recurrence of groups is actually something that was developed also elsewhere. So imagine this is a model that has a very minimal gene regulation system, and then cells can decide what to do based on the environment they find themselves in. And this environment is set to switch between two different environments, 1 and 2. And then this regulation can evolve in different ways, so cells can stick or not stick, and so on and so forth. So again, the time scale of this environmental change is set a priori. So this is a model, but the idea is that, for instance, I don't know, lunar cycles, day-night cycles, these are important in order to give the pace of multicellular, the evolution of multicellularity. So what we started to think is actually, do we need necessarily this kind of external setting, or can we actually have something like in the model of the Cheese-Effers propagules that is somehow inherent to the ecology of the system? And to do this, we started thinking like, so this paper was presented yesterday in the last talk by Professor Datta. And it's a paper where they thought, okay, what if I couple the replicator dynamics that is typically used in game theory to another equation that is describing the variation of the environment? And the payoffs of my payoff matrix for the replicator dynamics will depend on the environment, and vice versa. The environment will depend somehow on what strategies are there in the game. And so when they did this with like the prisoner's dilemma game, they ended up having these oscillations. That, as I said before, these are center kind of oscillations. They are oscillations that do not have really a known period. They are not really realistic. I mean, it was interesting to see that you can get this oscillatory dynamics by coupling the environment and the game. But yeah, there's a number of things that are not completely clear in this kind of model. Also, there is a sort of how to say, the identity between payoff and fitness is not completely clear. There are other papers that have done this a bit more precisely. So Leonardo Miele, who was a master's student a few years back, and he's now a postdoc in Torino, wanted to try to apply this method to try and see whether we could obtain some cycles that look like life cycles by coupling some feature that is some feature of single cells to their payoff through an environment. So the model he came up with is a model where you have resources, so these little dots, and then you have cells of two types. You have fast and slow cells. And the hypothesis is that fast cells are better in foraging locally, but if they are within a group, they will end up being in the front, and this is where cells die, you know, in dictyostelium. So they will propel the group, carrying also slow cells, but then they will die. On the other hand, the slow cells will be very bad in eating when they're alone, because they can't go sufficiently fast to catch the bacteria. On the other hand, they will exploit fast cells when they are in a group. So the fact of being fast or slow has two different implications on fitness if cells are alone or if they are grouped. Okay, we can translate this into payoffs. By simplicity, we took a payoff 0 for slow cells outside and payoff 0 for fast cells in groups, but that can be relaxed. And then we also assume that the probability of being isolated or being grouped depends on the availability of resources. So if resources are plentiful, then you would rather go alone, otherwise you would rather aggregate. So this can be put into the same kind of equation as the one by Joshua Weiss and colleagues, but this time the environment is actually given by this equation for the resource that has a logistic growth and that is used up at the same rate by both kind of cells, both slow and fast. What is nice here is that there is a strict correspondence between the payoff and fitness. So the growth rate of the population of cells strictly corresponds to the average payoff in the replicator equation. And this payoff is computed like this. So this system exhibits under certain conditions oscillations that look very much like the life cycle of dictyostelium. So when resources start to decrease because they are used up by the population of cells that are increasing in their number, then the proportion of fast cells increases. But when they are very numerous fast cells, so they start being exploited by slow cells, and then they will decrease. So this decreasing success of groups translates in the fact that resources can start again growing up. And then this increase of resources will also cause an increase in the population size, and so on and so forth. So the cycle starts again. So these cycles are observed in a large region of the parameter space, and notably when the two key parameters of this system, so the payoff of fast cells when they are alone and the payoff of slow cells when they are in groups, are high. They are also associated with some specific period or frequency. Now, the question we were asking is, what is the reason a system would find itself here, actually? Why should cells start being very exploitative from the beginning when groups are not there? So Leonardo used another approach that is adaptive dynamics, as we saw yesterday. And he said, okay, let's consider that lambda S, so the level of exploitation of fast cells by slow cells within a group evolves over time. So this is a continuous trait. At the beginning, we can imagine that cells do not exploit other cells. They're just, they can coexist at equilibrium when this lambda S is small. And then let's calculate what is the invasion fitness, as I was explaining yesterday. So what is the fitness of a mutant, a rare mutant in a population of residents? Okay, the resident trait is this level of exploitation, and this is a mutant level of exploitation. And now when we can compute, in this case, analytically, this invasion fitness, and it turns out that this invasion fitness is always positive when the exploitation increases. So this is somehow intuitive. Higher levels of exploitation will evolve. Okay? A nice feature is also that it will take longer and longer for them to evolve as exploitation is more extreme. So somehow there is this diminishing return of exploitation that was not put in at the beginning, and which means that when you have a lot of exploitation, actually you might have multiple different types coexisting. So the timescale separation between mutation and substitution could not be valid anymore. So that could be also an explanation of why there are many different types in nature. Okay, so what does that mean concretely is that if you start from an equilibrium where fast and slow cells were coexisting at equilibrium, so there was a certain proportion of slow, a certain proportion of fast cells, and now you are progressively increasing because of selective pressures, you progressively increase exploitation, the system goes naturally towards oscillations. So you have the emergence of like a proto-life cycle where certain cells will gain in the aggregated phase and some other cells will gain in the loners phase. So you have a natural compartmentalization of social strategies that resolves naturally the conflict between them, because there's no more conflict if, you know, cells are doing different things at different times. So I like this work very much. It's not cited by anybody. It's, I don't know, people don't like it, but I think it's a nice idea that you have a way to naturally evolve like a frequency that is the collective return in a life cycle. All right, and now we can go to the questions. Thank you. Yes.
So your last part was a bit fast, right? So what determines this the frequency? Again, can you give some intuition, like an intuitive explanation of how you drive this, how this oscillation would
Yes.
emerge?
Yeah. So as conflicts become larger and larger within groups, then the solution is to compartmentalize them. So you have a part of the life cycle where you have exploitation within groups, and a part of the life cycle where cells are free living. And since there is this pleiotropy of, so the trait is speed, and this trait is pleiotropic, so it's good to be fast when you are alone, but it's bad to be fast when you are in a group. So then, on average, both types end up coexisting, just because you know, they get their payoffs at different times. So this is, you know, mathematically comes up like a hot bifurcation in this system. And, you know, I think this is a general feature of this kind of equations when you have, you know, resource, consumer, and then the evolutionary part is actually reflecting the growth rate of the population. So I'm ready to bet that you can create many models of this kind with different kind of hypotheses. I think the key hypothesis is this trade-off between what you gain when you are outside or when you are inside groups.
So when you talked about those motile cells, you said that some of them are slow and some of them are fast, right? So how slow you will call it slow and how fast you will call them fast?
Yeah, so in this model, there's nothing is quantitative, is purely qualitative, is not even mean field, is you know, some kind of, there's no microscopic model that is, that is quantified here. But it's something we would like to do. For instance, we could do the same with these individual-based models and see whether this holds. I think actually, I started thinking about this. You probably, if you have an individual-based model, you probably don't need the dependence on the resource itself, because when you increase density, as I was showing in the simulations before, you have naturally increased aggregation. and also when you increase speed, if speed is not too large. So you probably can get that, when you have a large number of fast particles, then you will naturally form aggregates.
Hi, very interesting talk. So I was intrigued by the observation of bimodality in motility that you found. Just a comment, we find similar bimodality also in fish motility. But not all species. Some species show this, very clear bimodal structure. So I would be curious to talk a bit more on how that impacts the collective behavior.
Yeah, I think this is very interesting, and has not been studied a lot because in previous studies in Dictyostelium, for instance, since this is not a high percentage of cells, people thought it was an artifact, and they just threw them out and kept the main motility pattern. Thank you.